Are people joining? Yeah. So, good morning, good afternoon, everybody. Welcome to the Encardia's webinar on iPSC solutions for gene therapy. We've just started, so we're just going to give it 1 to 2 minutes, allowing everybody to join, and then we can get started. We'll just give it 1 more minute, and then we will get started. Oh yeah, I forgot to mention. So there are, I guess, tabs for Q&A and for chat. So if you have any questions, please type them in there. I won't be able to look at them during the webinar, but at the end I will take a look, and if there are questions that we're able to answer, we will do so. And if we're not able to answer them immediately, we'll touch base with you after the webinar to follow up. Okay, we'll get started. So today I'd like to present a couple of case studies where Encardia supported various clients in delivering iPSC solutions for gene therapy. And the whole premise of this is that we need to be using human models as early as possible in your pipeline, not only to ensure that the candidates you're working with are safe, but also that they are able to rescue these functional deficits that we see in the disease lines. So just a bit about myself. So I've been with Encardia for a number of years, over 4 years now, and I lead the drug discovery team, so supporting projects, both from a scientific and technical perspective, mainly across the neuroscience space, but also, as you would guess, the cardiac space as well. And my background is very much in drug discovery across a whole range of different drug modalities and disease indications. So the webinar is split up into essentially 3 sections. So this is kind of an overview of how we think gene therapy and iPSC technology are almost a perfect match for one another, and try to explain how iPSC tech can de-risk and accelerate your gene therapy program. And then I'll delve into 2 to 3 case studies where we specifically used both iPSC-derived cells in combination with the AAVs at various stages of that drug discovery process. And then at the end, we'll just hopefully nicely summarize how we think, again, iPSC-derived cells can de-risk gene therapy development. So, for gene therapy, strategies can be delivered either ex vivo or in vivo. So the ex vivo is depicted on the left and the in vivo on the right. So essentially for the ex vivo approach, patient cells are collected, genetically modified outside the body, expanded, and then returned back to the patient. Kind of the added value of ex vivo, it gives you much more control of actually what you're delivering back to the patient. So the steps involved in modification of those of those cell types, the expansion can all involve significant QC to ensure what is delivered back to the patient is both safe and functional. And in vivo approach, the genetic material is delivered directly to the patient, and I know this space is accelerating at a tremendous rate. And a whole range of different modalities can be used to deliver that genetic material. So that is either using viral vectors, such as AAVs, that can be lengthy, but that is not so common, but also non-viral means, such as lipid nanoparticles. One thing to emphasize here is that regardless of the approach, the goal is the same, is to deliver the therapeutic gene safely and effectively. And this makes the robust analytical testing essential throughout the development of the gene therapy. So when you're developing a gene therapy platform and mapping out your path to get to a patient, there are three important pillars, release test pillars. So that is the identity, the biosafety, and potency. And these are applied across all steps during that manufacturing process until it's ultimately delivered to the patient. So this ensures that all the raw materials, so the plasmids, the cells, and all these different components undergo rigorous QC to ensure that they pose as little risk as possible to patients. The manufacturing process itself is strictly controlled and heavily monitored to indeed ensure that the cells that are produced or the virus is free of contaminants, and then ultimately the final drug product, so ensuring that it is not only safe but it is also stable, for example. You'll notice in the slides we've also highlighted potency throughout the process as well. And the reason for highlighting that is that potency ultimately links product quality to biological activity, and hopefully that is then, informative or predictive of clinical performance. And it's important to realize that regulators expect sponsors, so the developers of these therapies, to not only demonstrate that the product is present and it's safe, but it also performs to its intended function. So that's why I'm putting quite a bit of emphasis on potency, as it serves as a bridge between the manufacturing and, again, as I mentioned, predictive of the patient outcomes. So regulatory agencies define potency as the product's ability to produce the intended biological effect. And something as complex as gene therapies or other genetic modifications, there is probably not a single assay that can fully capture all different aspects that need to be monitored associated with that therapy. Thus, developers normally use a matrix approach, which we've tried to summarize on the right here. And just to highlight, it's not just one regulator that is saying it, it is multiple regulatory agencies that are emphasizing the need for this type of approach. So in the early stages of your development program, it's important to quantify the infectious titer, but also capsid titer. And then very quickly from there, moving into expression of your mRNA. So really, what is your gene therapy being expressed, both at the mRNA level and also at the protein? And then ultimately moving into, should we say, these more complex assays, functional assays, to show that the gene therapy product is not only expressed, but it can then also rescue these functional deficit. And it's important to realize that we can have the best intentions at the beginning of any gene therapy program, but as the therapy itself begins to develop and to evolve across your program, so must the assays to fit and become fit for purpose as well. So, in terms of I guess, the relevance of the assays to use both for, I guess, for all the stages of your program. It's important to pick the most relevant models possible to give you to help you make informed decisions as you go through that process. So this was nicely summarized in a paper back from 2015, and essentially what it's saying is that in a perfect world, we would use, the model that is closest to the disease that you're trying to treat, and ultimately, we've got all going for human diseases. It's ideally a human iPSC-derived model, and in a perfect world, if when you take that iPSC cell from a patient and differentiate it to the relevant cell type, it then has the disease phenotype without endogenously present, without the need to add on additional stimuli. And then ultimately, you want to be able to demonstrate that both from a molecular perspective, but also from a functional perspective, your therapy can then rescue that disease manifestation in the iPSC-derived cells. So the synergy between iPSC and gene therapy. So iPSCs, since they were discovered, the technology has come advanced quite significantly. And one of the added values of one of the many added values of iPSC-derived cells is that not only do they retain the genetic background of the patient, so really the patient population that you're trying to treat, they are almost provided indefinite supply of cells for your gene therapy program. So we know a lot of these drug discovery processes are complex and lengthy, so often running from 3 to 5 years. So using that patient-derived cell and then differentiating it to your cell type at scale ensures that you have sufficient cells for your entire discovery phases, but also then the same cells can then be used in a GMP or GXP environment for release testing of your product. Kind of the differentiated cells or the cells differentiated from iPSCs can often have functional readouts. So for cardiac cells, we look not only at the beat rate, the field potential, so really the functionality of these cardiomyocytes. And then if we move into the neuronal space, we can also begin looking at synaptic activity, as well as calcium activity, for example. And these types of readouts are either not possible in other cell models, so immortalized cell lines, it's not possible to get this level or this degree of functionality. So really gives you confidence that if your gene therapy is able to modulate a functional readout, it's going to do the same in a patient. And then ultimately, at least from Encardi's perspective, we not only have the expertise in, reprogramming primary cells to iPSCs and differentiate them to the relevant cell type. We can also work with you to be able to support the development of the assays, which will not only be used in that discovery phase, but also think with you what type of assays can then be transferred into that GXP environment, ultimately for a release testing. And kind of where we will walk you through is the manufacturing of the cellular models, how we've used these models to quantify transduction and evaluate vector toxicity, and then moving into how we've used both the iPSC-derived cells from a patient population to assess AAV efficacy, and then ultimately how these assays can then be transferred into a GMP environment. So one of the first steps that it's important to evaluate is, are the relevant cell types able to take up your vector, and are they then able to subsequently express your gene therapy, and then ultimately is the protein of that gene therapy one expressed, but also is it localized correctly intracellularly. And this is a case study that we generated a couple of years ago, where we actually took iPSC-derived cardiomyocytes and we transduced them with AAV from at least 6, potentially 7 different serotypes, and the AAVs were expressing in GFP. So this allowed us to quantify both transduction efficiency into the cardiomyocytes, but using a high content platform, we not only looked at transduction efficiency, but we also looked at cell viability as determined by the number of nuclei. And what we can clearly see is that there are some serotypes which are able to very effectively transduce our iPSC-derived cardiomyocytes, whereas there are other AAVs, for example, that are not. And at virtually all the concentrations we tested, we saw minimal toxicity associated with the AAV. So at least from that perspective, it gives confidence that if you do hit the right cell type, we can effectively get the gene therapy into the cell type, and that there should be the minimal toxicity, at least for that very specific cell type. Kind of moving on from that, once we get the therapy into the cell, we need to ensure that the therapy itself is going to be expressed. So what we have on the left here is with those cardiomyocytes, after transduction, they were lysed and a qPCR performed for their target gene expression. And what we can nicely observe is that in a MOI-dependent fashion, we can see an increase in target gene expression with AAV2 clearly outperforming AAV1. And then again, using high content imaging, we did ICC, immunocytochemistry, for the target protein, just to ensure that the protein itself was being expressed, and it was then correctly localized to the correct subcellular compartment. So kind of moving into a more extensive case study where we did it with a client, and we were evaluating their AAV in the cardiomyocyte disease model. So ultimately, the AAV wanted to be used long-term, as I mentioned, not only for these early discovery phases, but also for later for batch release testing. And kind of one of the, I guess this speaks to my earlier point about, an indefinite source of cells. So what we have here are our Insight cardiomyocytes, which we differentiate in bioreactors, which is an image shown in A. And the bioreactor differentiation that we have has kind of two advantages. It produces quite a high pure population of cardiomyocytes as determined by troponin T. The cardiomyocytes themselves are functional, as seen by a typical ventricular-like action potential when quantified on multi-electrode array. But also, because we can use bioreactors, it makes the entire process scalable. So we can begin to produce billions of cells in a single run. And what we've done internally, at least, is we have stored these cells for, I think, almost 5 years. And what we see is that there is very little change in both the purity of the population that is produced. The postal viability remains virtually identical, and their performance on function, as assessed by functionality, is consistent both from batch to batch, but also across time. So as I mentioned, this whole process of producing functionally relevant cells is highly scalable. So, kind of the disease model we were looking at was Friedreich's ataxia. So, it's characterized by both neurological and cardiac complications, and ultimately it's a genetic disorder driven by an expansion of a trinucleotide repeat in the frataxin gene. And ultimately, this leads to lower levels of frataxin, and then this can have a number have a number of consequences, mainly abnormal calcium handling and mitochondrial dysfunction, which then ultimately leads to cardiac hypertrophy, impaired contraction, and it's quite a devastating disease. So it needs some effort behind it to try and resolve those issues. So essentially, together with our partner, we designed a workflow for this entire process from end to end. So this was sourcing of the iPSCs, mainly with the Friedreich's ataxia expansion, the optimization of the differentiation of the iPSC into cardiac cells, both initially in a low throughput format, but once the process had been optimized, we scaled it to a bioreactor. We developed the assays to quantify various aspects of the dysfunction that we saw or that we expected to see in the disease. And then ultimately, can we then modulate that disease phenotype both from a molecular and functional perspective, and can that AAV then rescue that disease phenotype? So what we have here is the iPSC-derived cardiomyocytes. So in A, we first wanted to quantify the levels of frataxin in those cardiomyocytes, as we'd expect that in the mutant line, we should see low levels of frataxin. And indeed, that's exactly what we see here. So my colors, I'm not very good with the colors, but in the orange color is the control line, and in the dark red is the mutant line. And in both, say, culture media, so in baseline or supplemented with iron, we can see that frataxin levels are significantly reduced relative to the controls. And in B, we also ran a FACS assay, so looking at cellular ROS. So what we can see is, again, in the control population, there is no real increase in ROS production. However, when we look at the FRDA line, we can see that at least in at the higher levels, we can see significantly increase in ROS production. And then probably more relevant for from the calcium handling perspective, what we can see here is that in A, in the upper plot, we have a wild-type cardiomyocyte where there is spontaneous calcium activity, transient peaks, with the typical peak form that you would expect from these cardiomyocytes, whereas in the Friedreich's ataxia, the frequency of these spontaneous activity is significantly reduced, and also the PW80 is significantly increased as well. So the next question is, kind of very similar to what I was mentioning before. We have to test if the AAV was entering the cardiomyocytes, and if it was entering the cardiomyocytes, was it able to then express frataxin? In this instance, we purely looked at the frataxin protein, and then ultimately, can the expression of frataxin, so trying to replace the loss of frataxin, can it then lead to reduction in ROS levels, but also functional rescue of this calcium handling? So what we can see is that in A, when we transduced the cardiomyocytes with the AAV of frataxin, we got a significantly higher expression of frataxin relative to the controls. Probably these levels are going to be a bit too high and actually may cause some damage. So we can always begin to optimize the transduction by altering the MOI to bring the expression levels of that frataxin down to appropriate levels. And what we can see is that in B and C, the transduction with the AAV was able to significantly reduce the PWD80 in the Friedreich's ataxia line, and also be able to reduce ROS levels. So at least from a confidence building perspective, there was confidence that the therapy itself was able to rescue disease-associated phenotypes in a disease, in a physiologically relevant cell model system. Kind of switching gears from cardiomyocytes to the neuronal space, so really for ALS. Again, similar to Friedreich's ataxia, devastating disorder. And ultimately, at the moment, there is no cure for this disorder. And that needs to change. So ultimately, ALS, there are a number of different genetic causes for ALS, but one of the key proteins involved in the disease pathogenesis is a protein called TDP43. And there are a number of molecular events which ultimately lead to neuronal dysfunction in the pathogenesis, at least associated with TDP43 mutations. So initially, TDP43 mislocalizes from the nucleus to the cytoplasm, where it subsequently aggregates. TDP43 is a protein involved in RNA splicing of hundreds of different transcripts. And one of those transcripts is stathmin-2, where if TDP43 is not present, stathmin-2 is misspliced and you get a truncated species. And as I mentioned, all of these molecular deficits collectively work together to then ultimately lead to neuronal dysfunction. So what we did is we built a suite of assays to measure different aspects of the of that disease, of the molecular etiology of the disease. So we used a high-content imaging platform to look at TDP43 mislocalization, HDRF assay to look at aggregation, and then qPCR to look at missplicing. But then we bolted on different readouts to not only give us more information about how the therapy could be rescuing the disease phenotypes, but also to bring confidence that the therapy itself was not going to be toxic. So we included parameters such as neuron outgrowth, NFL release, and then also quite relevant electrophysiology, as I mentioned, to ensure that the therapy itself was not interfering with neuronal function. So this is just an example of one of the data, of some of the data. So what we have here in A are iPSC-derived motor neurons labeled for nuclei, so DAPI and then TDP43. And what we can see is that when we treat these iPSC-derived motor neurons with a stressor, TDP43 mislocalizes from the nucleus to the cytoplasm. And I think this is an important point to stress. In one of my earlier slides, I mentioned kind of the gold standard would be to have a human iPSC-derived model to use in your drug discovery program. But in a perfect world, we would have a disease patient line which shows the disease deficits without any stimuli. For some diseases, so as we saw for French's ataxia, we were able to do that. But in the instance of ALS, at least some of the patient lines that we've used, we've not observed a natural disease phenotype in these molecular readouts, hence our need to use a stressor. And kind of accompanying this mislocalization from the nucleus to the cytoplasm, we can also see that co-commitment increase in TDP43 aggregation as quantified by HDRF. So again, with taking the same route as we always have done, is assessing if the therapy itself can go into that physiologically relevant cell type. So on the left here, we have these iPSC-derived motor neurons, in this instance in a co-culture with astrocytes. And we transduced them with the virus, but encoding GFP. And what you can greatly appreciate is that after 2 weeks post-transduction, we get high levels of expression of GFP, indicating that the virus can enter the cells. And then ultimately, with this therapy, we saw that the therapy itself was able to significantly and effectively inhibit TDP43 aggregation. So it almost caused a 75% decrease in TDP43 aggregation. Ultimately, this would then support, okay, this is the first step. Can the therapy then rescue the other deficits associated with ALS? So as I mentioned, the missplicing of stathmin-2 and also the electrophysiology deficits that we observe in the mutant line. So the key takeaways. So how can iPSC-derived cells de-risk gene therapy development? So iPSC-derived cells can be differentiated at scale. iPSCs can be sourced from patient populations, so really from patients with the disease that you are trying to go after, and they can then be differentiated at scale to the physiological relevant cell type, and quite often with a high degree of functionality. This then allows you to, during your screening phases, but also through your later stages and also through your batch release testing, enables you to look at both molecular and functional readouts to see if the therapy itself is able to reverse some of those functional deficits. And as I mentioned, the platform that's developed for the early stages of the program can also then be transferred for AAV release testing. And ultimately, what you're doing is you're bringing confidence at an early stage that your therapy is able to rescue disease phenotypes. So ultimately, all those slides, and I've tried to summarize it onto one. So the models that we have available can be used and adapted to look at both transduction efficiency as well as target mRNA and protein expression, and then they can actually be used to model the disease and actually determine if the therapy itself can rescue or pathophysiological specific functionality. Thank you. If there are any questions, please ask them. So I have a question. At what stage of AAV development do you see the biggest value for iPSC-based models, vector selection, potency, and clinical translation? So I think here it's going to be at all stages. So you have the iPSC-derived model, so this very specific cell type that you're trying to transduce. So it enables you to select the vectors which can transduce that specific cell type most effectively. Because you can produce the cells at scale, and they can then be frozen, it enables potency testing. One of the challenges, I think, in any cell-based assay is variation. So being able to have that one model which is frozen down and validated, which allows you to then, develop your potency assay, but then use that potency testing to support both optimization and then release of your therapies is crucial. And then, clinical translation, it's a human model. It may not have the complexities of an in vivo animal model, but having that human component, I think, is indispensable to any drug discovery program. Same person. What are the biggest barriers preventing broader adoption of iPSC-based potency assays in gene therapy programs? I think one of the challenges is, and I mentioned this specifically during the ALS, is getting that disease phenotype in this iPSC-derived cell. It's not always so easy, and it may require quite some time to be able to get that functional, both the molecular and the functional deficit in those disease models. So I think there, being able to work with a partner at speed, because speed is important for all these diseases, is crucial to make sure that we can support you in using iPSC technologies and helping you de-risk their usage, by not, by ensuring that the cells and the models and the assays that are used are the correct ones, with the experts next to you. The iPSC-derived cells are comparable to the wild-type cells. I'm not sure I fully understand your question. The iPSC-derived cells are comparable to the wild types of cells for the AAV intaking. They should. Okay, and please shout out if I've not answered your question correctly. They should be. So when we do AAV uptake or transduction assessments and expression, we have a device. We can use. Are the receptors similar to the AAV entry? They should be. So, with the case studies that we saw with the Friedreich ataxia and with the ALS program, the client's therapies were able to very effectively enter those cell types. I'm pretty sure that if we start moving into more exotic AAVs, there will be some receptors which are not expressed on our cell types. So there, I think it will be important to understand, if you know how your AAV is entering, if that indeed our cell types express those receptors. Okay, I don't see any questions, but if you have follow-up questions as you're thinking about this later, please don't hesitate to reach out to Incardia, and then we'll be happy to provide you answers as well. So lastly, I'd like to thank you for all joining, and I wish you a nice rest of the day.